Dr. D, 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 Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be finishing up Chapter 4 of our Bauman textbook, talking about staining and classification of these microorganisms. So with staining, remember the point of staining in microbiology is to increase contrast. If you're looking at E. coli under the microscope and you haven't stained them, well, they're going to be very difficult to spot because they're clear, they're colorless, and looking under the microscope, you're really going to have a tough time seeing E. coli. So what we do staining procedures which introduce dyes, and these dyes stick to the E. coli, and therefore you can see the colorful E. coli. You can stain them a variety of colors. There are pink stains, purple stains, green stains, blue stains. There's a myriad of different stains we can use. So we have to understand that staining in general is done so that you can increase contrast and you can see parts or all of the cells. And the staining procedures have to be done a certain way. So we need to learn about staining as well. So again, coloring a specimen with stain increases contrast mainly, but also it can help with resolution. But first, specimens must be prepared for staining. So how do we typically prepare specimens in the microbiology lab? Well, it's called a smear technique. You take bacterium from an inoculum on a loop, you spread the bacteria in a drop of water or half a drop of water, and then you allow this solution, basically it's a suspension of bacteria, to dry, okay, air dry. So now you've got a scattering of bacteria in this smear. Now, here's the trick. You don't want to just go ahead and stain at this point, because if you tried to stain, the bacteria really aren't well stuck to the glass, and that they would just wash right off the glass. Does that make sense? And that's why we do the next step here. It's called heat fix, where you pass the slide really quickly across a flame. Remember in aseptic technique, we discussed that a little bit there. When you're doing aseptic technique, you want to pass that across the flame. So you're going to pass the slide across the flame, and this will do one main thing. All right, this will adhere the bacteria to the slide. Heat fixing helps to adhere the bacteria to the slide. Therefore, when you go to do your staining, the bacteria don't simply wash off the slide. So let's talk about some of these principles of staining. First of all, you need to know that we use various dyes, and these are usually salts. And we either use acidic dyes or basic dyes. Okay, typically, I mean, you could use some non-polar dyes, but those are special application. In microbiology lab, we typically use acidic dyes or basic dyes. Acidic dyes stain alkaline structures. So what does that mean? Okay, here's what I need you to understand. Acidic dyes have a negative charge. Okay, you need to know that acidic dyes have a negative charge so that they stick to and stain alkaline structures in the cell. You know what that means? Structures in the cell that have a positive charge. Did you guys follow that? The dye, acidic dye, is a negative charged dye. They stain alkaline structures. Here's something I want you guys to understand. This will help you so much in the future, y'all. Okay, listen to this. This is going to help you a lot, okay? This was something that took me a while to understand when I was, back in college. And that is this. Acidic substances, when you're talking to a biologist or a biochemist or a molecular biologist or even chemists, when they're talking about acidic substances, they're talking about negatively charged substances, right? So when you hear acidic, think negatively charged, okay? This will help you so much. And positively charged substances are basic or alkaline. Did you know, again, that basic means alkaline? So basic or alkaline structures or dyes are positively charged. Acidic are negatively charged. And remember how charges work? Opposites attract. So if you have a negatively charged dye, it's going to want to stick to positively charged molecules, right? Or specimens. If you have a positively charged dye, it will stick to negatively charged specimens, okay? So this is really important to understand. Now look at this. This is something else you should really understand. Look at this. More common since most cells are negatively charged. Look what it says here. Most cells are negatively charged. Have you ever heard of membrane potential? I think we talked a little bit about membrane potential. Membrane potential is the charge difference across a membrane of a cell. It's a voltage difference across the membrane of a cell. Remember, most cells have a negative 60 or 70 millivolt membrane potential across a cell. This means that most cells have a negative charge to them. So what kind of dye do you think you'd want to use to stick to a typical cell? The cell is negatively charged. So yes, you would not want to use an acidic dye because acidic dyes are negatively charged. Does that make sense? The cell is negatively charged at the surface. The dye can't be negatively charged because it won't stick to the cell. Does that make sense? So I wouldn't want to, like, let's say if I'm trying to stain E. coli, I wouldn't use an acidic dye because I know my E. coli have a negative charge. I'd want to use a basic dye. Does that make sense? Because basic dyes or alkaline dyes, these dyes have a positive charge. So most dyes that you are going to use in the microbiology lab, most of those dyes are basic dyes. Okay, and that's true. Most of the dyes that we use are basic dyes. Basic dyes have positively charged groups and bind to negatively charged molecules such as DNA, RNA, a lot of proteins, the surface of bacterial and archaeal cells. So look at this. Basic dyes are very, very, very useful in the microbiology lab because basic dyes are positively charged, and bacteria and archaea that we study are negatively charged, and a lot of the molecules inside are negatively charged. On the other hand, acidic dyes are still useful, but they're not going to stain the whole cell usually. They will stain specifically negatively charged portions of the cell. Does that make sense? Sorry, they will stain positively charged portions of the cell. I almost got myself backwards. So acidic dyes will not stain the whole cell usually, but they do stain anything in the cell that's positively charged. Does that make sense? So let's talk about the staining itself. What types of staining are there? Well, the most simple stain is called a simple stain. And these are when you use one single basic dye. For example, crystal violet is a very common purple dye we use. It's a basic dye. Safranin, which is a reddish pink dye. Or methylene blue, which as its name suggests is a blue dye. These are all basic dyes and very common dyes that you will come across in microbiology lab. All of them have a positive charge, so they're great for attaching to your negatively charged E. coli, for instance. And why do we stain? Again, for contrast. And also so that we can see the size of the cell. We can see the shape and arrangement of the cell. Do you guys remember the difference between shape and arrangement? We talked about this before. Shape is the actual shape of each cell. You could have coccus-shaped, bacillus-shaped, right? Spirillum-shaped. That's shape. Arrangement means how are these coccus aligned, or how are these bacillus aligned? Are they strepto or staphyla? Strepto means chains of. Staphyla means clusters of, etc., etc. So look here. Here's some simple dyes. It looks like a methylene blue dye on the left. See how you can see these coccus-shaped cells? And look here, lighter, you can see some bacillus-shaped cells as well. Look at this. Bacillus cells here, 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 and coccus shaped here, 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 here. Great. But notice, even though this is two different species of cells, it looks like two different species. For example, the rod-shaped ones might be E. coli, and the dots may be Staphylococcus or something. Now, it just stains all the cells. A simple stain stains all the cells, so that what can you tell? You can only tell shape and arrangement. Now look here. Again, this is a stain with crystal violet. I can see coccus-shaped cells, and I can see rod-shaped cells. These are very likely two different species, but it doesn't determine what species they are. It doesn't differentiate between them. It just stains everything purple. Okay. Now, if you do want to differentiate between different types or different species, you'd want to use a differential stain. And this is when you use more than one dye. Okay. And usually the dyes are different colors. Usually the dyes are different colors. So you could use a purple dye, and then later in the protocol use a pink dye, such as the gram stain procedure. You can use a green dye followed by a purple dye. So there are many different procedures that you can use. This distinguishes between different cells, chemicals, or structures. So here's some examples of these differential stains, the stains that have two different colors, right? Gram stain, acid-fast stain, endospore stain, and other histological stains. So one staining protocol that you should be well aware of in any microbiology class is called the gram stain procedure. Remember, we learned in a previous chapter that there are two different types of cell wall for bacteria. There are bacteria with gram-positive cell walls, and there are bacteria with gram-negative cell walls. Remember that? Well, the gram stain procedure was developed by Christian Gram, a medical student back in the 1800s. And what he showed was that you could use a technique of staining with two different dyes. Crystal violet dye, which is a purple dye, and safranin dye, which is a reddish pink dye. And at the end of this staining procedure, you can tell which cells are gram positive. That means have the gram positive cell wall type, and which cells are gram negative, meaning they have the gram negative cell wall type. So what's the protocol? The first step is to add crystal violet, which is a purple dye, onto the cells for 1 minute, then rinse. Now look, all cells are stained purple. Why do you think all cells are stained purple? Well, because crystal violet is a basic dye. Basic dyes stain all cells. Okay, so let's say I have two different species of bacteria, everything will be purple. Even if some of these bacteria are gram negative, even if some of these bacteria are gram positive, all of these cells will appear purple. Does that make sense? But that's only step one of the procedure. What do you do next? The slide is flooded with iodine. Do you know why we use iodine? Iodine acts as a mordant. I need you guys to understand what the term mordant means. Iodine in this case is not a dye. Iodine is used as a mordant. Mordants help fix a dye in place. What really happens here is that there is a crystal violet iodine complex that forms. So crystal violet and iodine complex and become bigger. And this reduces the permeability of crystal violet. What does that mean? It makes it harder for the crystal violet to get washed out of the cells. Okay? Now, so the cells at this point, the cells are still purple. What do you do next? The slide is then rinsed with a solution of ethanol and acetone. Okay, this is called Graham's alcohol. Ethanol and acetone for 10 to 30 seconds, and then rinsed with water. Do you know what this did? The ethanol and acetone solution serves as a decolorizer. A decolorizer. What do you think a decolorizer does? That's right. It washes the dye out of the cell. But guess what? Here's where the differential part of this differential stain process works. All the stains, all the cells, I should say, stained purple, right? All the cells stained purple. And now you're adding ethanol and acetone to wash the purple out of the cells. However, notice how you only do it for 30 seconds. You want to know why you only do it for 30 seconds? Well, because by 30 seconds, the gram-negative cells have completely got washed clear, but the gram-positive cells have only partially become washed clear. Does that make sense? Did you guys follow me? Do you remember how gram-positives have a really thick peptidoglycan cell wall? You guys remember that? Gram-positive cells have a very thick peptidoglycan cell wall. Well, that makes it harder for the iodine crystal violet to get washed out of the cell. Does that make sense? But gram-negatives don't have that really thick cell wall. So it is really easy to wash that stuff out of the cell. You see what I'm saying? So anyway, at this point, if you were to look under the microscope, you see these purple cells? These are gram-positive cells. Where are the gram-negative cells? Where do you think the gram-negative cells are? That's right, they're clear. You can't see them. They're colorless. You can't see them. So guess what the last step is? The last step is to add what's known as a counter stain. This means color number 2. You flood the cell with safranin. This is another basic dye that's like a pink or a red for 1 minute. And then you rinse and blot dry. Look what you have now. Look what you have now. The gram-positive cells remained purple because the purple never washed out fully. But the gram-negative cells are a light pink. So everywhere you see light pink, that's a gram-negative cell. Everywhere you see purple, that's a gram-positive cell. You see how this is a differential stain? It differentiates between different types of cells. And do you see how this might be useful when you're trying to identify bacteria under a microscope? All right, so that's very, very interesting stuff. Here's another example of a differential stain. It's called the acid-fast stain. We're going to be learning about this in a lab module. Here's another example. This is called the spore stain or the endospore stain. We learn about this in a lab module as well. These little green dots are the endospores that this particular bacteria, Bacillus anthracis, can make. Now in pink, you see this pink here? This pink is called the vegetative cell. The vegetative cell is the bacteria itself. It's the living bacteria. But these bacteria, the Bacillus or the Clostridium genus of bacteria, are able to make endospores so that they can resist harsh environments. Does that make sense? So the bacteria itself is pink, but the endospore it's forming is green. And only 2 genus of bacteria can do this trick, can make endospores. Only bacteria in the genus Bacillus, only bacteria in the genus Bacillus, and bacteria in the genus Clostridium. Those are the only 2 genus of bacteria known to make endospores. And I also mentioned earlier that there are histological stains as well that differentiate between types of cells. You don't need to know a lot about this, but typically when you move on to throughout your nursing program, you'll see that H&E stain is a very, very common type of staining, hematoxylin eosin staining. And that's what you'll see in a lot of histology books where you look at tissue samples. You know those tissues that are slightly pink, slightly purple, and they're showing you sections through a heart or something like that. They're typically stained with hematoxylin eosin. All right, so let's take a quick break time with Tig. And we'll come back. We'll continue talking about staining, and then we'll finish up this chapter. I'll see you guys in just a minute. All right, everyone, welcome back. So where we left off, we're about to talk about special stains. These are special staining techniques that bring out contrast of what you're looking for. For example, if you're looking for flagella, there are stains that will allow you to see those fine whip-like structures. If you're trying to see things that cannot be stained, such as non-polar structures of a cell, you could use what's known as a negative staining technique. You could use fluorescent stains as well. So let's talk about a few of these. Here's an example of a negative stain. Remember we learned in a previous chapter that some bacteria have a capsule, which is part of the glycocalyx, and the capsule is non-ionic, which means it's not charged. The capsules are not charged. So how would you use a dye, a basic or acidic dye, to stain something that has no charge? Think about it. Can you use a basic dye, which is positively charged, to stain something that's non-polar, non-charged? No. Could you use an acidic dye to stain a structure that's not charged? No. So what you would do is a negative stain. What that means is you stain everything on the slide except for the structure that you want to see. And in that way, you reveal what you want to see. Look, in this case, it's a negative stain to look at Klebsiella pneumoniae. These are bacteria that have a capsule. When you stain, the bacteria itself is stained. Look at this. You see this rod-shaped bacteria? The bacteria itself is stained. The background is stained. So the glass is stained on this slide as well. See? The glass is stained. The specimen is stained. But there are these halos. You see these clearings around each bacteria? I call these halos. You see these halos around each of these bacteria? Well, guess what that is? That's where the non-ionic, uncharged capsule is. The capsule can't be stained with these ionic dyes, these basic or acidic dyes. However, you can use dyes to stain the cell itself and stain the background, right? So when you stain the cell and the background, the only thing that remains unstained is what you're looking for, which you can't stain directly, and that's the capsule, see? So this is called the negative staining technique. What does negative staining mean? Staining everything except the structure that you want to reveal. Here's flagellar stain, which, remember, this is another type of specialized stain. In flagella stain, it reveals the very fine flagella of bacteria. So this is Proteus vulgaris, a very motile bacteria. It's got several flagella. It's peritrichous, which means has flagella all the way around the bacteria cell. And you can see individual flagella that are revealed with this flagellar stain. Normal staining doesn't reveal flagella. Okay, so here are some overview tables of what we've been talking about. And the last few slides here are really a review of what we talked about earlier in part 1 of this chapter, and that is about electron microscopy. Remember that in electron microscopy, electrons replace light as the illuminating beam, as the electromagnetic radiation. The wavelength of an electron is much, much shorter than light. This is why you get so much more resolution. And this allows us to get so much more detail. Here's a nice table juxtaposing light microscopy and transmission electron microscopy, so that you know that with the light microscope, what magnification and resolution you can expect versus a transmission electron microscope, so on and so forth. Remember, we covered this earlier. And look here what I wrote at the bottom. This is very important. Wavelength of an electron in a transmission electron microscope can be as short as 2.5 picometers, as in picometers, as in 2.5 times 10 to the minus 12 meters. So the wavelength of an electron is about 100,000 times shorter than the wavelength of light. This means that you can get much, much better resolution with an electron because the electron has such a short wavelength. And remember, the electrons have to travel through a vacuum. Electrons can't travel through air. Air will actually absorb the electrons. And also, we're not using glass lenses, are we, with electron microscopy? We're using magnetic lenses, electromagnets. So remember, you've got an electron gun. The electrons are shot through to the viewing screen through the specimen, and it's the electromagnets that focus the electron beam on the specimen. And again, this is the vacuum chamber, and this is the type of sample you would see. Remember with TEM, transmission electron microscopy, look at this. You can see bacteria inside of a section. Remember TEM, you're sectioning the bacteria. They're all sectioned. And so you can see the inside of the cell. You see the nucleoid here. You can see individual ribosomes. You can see the photosynthetic membrane vesicles. You can see so much detail. And remember when I said for specimen preparation, you must cut very thin sections for TEM, for transmission electron microscopy. And then how do you stain for electron microscopy? Do you stain with dyes? Nope. Remember, you stain with dense metals, heavy metals. Okay. And you can still do cool tricks with the electron microscope. You can do negative staining with electron microscopy. Remember, this is where the heavy metals do not penetrate the specimen but render a dark background. So what you would end up with is a dark background coated in the dye, the metal dye, and then the light structures, the light structures with the microscopy would reveal the sample you're looking for. And you can also do shadowing. Shadowing is when the film, the metal is only on one side of the specimen so that it gives a shadowy effect. This is helpful to look at viruses, flagella, DNA, etc. So look at here. This is transmission electron microscopy. You see all this detail inside of the cell, the nucleoid, the cell wall, the septum. So this is a cell that's dividing into two. Down here you can see this is negative staining with electron microscopy of a virus, what's known as a bacteriophage, a virus that can infect bacteria. Notice how the virus is not stained, but the background is. That's a negative staining procedure. And then look here, to reveal the flagella on this bacteria, Proteus mirabilis, you can see there's been shadowing here, where the metals are on the bottom side of these flagella. Okay, and do you remember we talked earlier in part 1 about scanning electron microscopy as well? This is, you're also coating the sample with metal. You're also using electrons. However, this reveals a three-dimensional image of the sample where you see the topography. Remember the surface structure of the bacteria. Why? Because the electrons, the electron beam is scanned across the surface of the bacteria. Remember, in scanning electron microscopy, you do not section the cells. You leave the cells intact and you scan the top of the cells. So you can see the surface topography. So this is a picture of me. When I was back in graduate school, I was tinkering around with this fluorescence microscope. And so, you know, fun times. I had hours and hours of time with this microscope in the lab. But anyway, I hope you learned a lot about microscopy. I do love the field of microscopy. I think it's very intriguing. So I'd love to answer any of your questions that you have. Just leave them in the comment box below, and I'll catch you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.